In the sixth step of the data upload process, we get to uploading the actual measurement data. In order to demonstrate this effectively, I've started a brand new study, which is why we get to see this warning here that we need to go through the separate steps. I'm going to skip the publication. I'm going to call this study test study 1. I'm going to create a new project called test project 1. And then I'm going to simply reuse the entirety of study 1. This will create a new study, but it will reuse the experimental design. I'm going to do this so that we can actually go through the experimental design quickly and match it up with the data that we're going to see in the spreadsheet. I'm going to use this, click on next. In terms of microbial strains, we have 4 of them: Bacteroides, Balti, Roseburia, and Prevotella copri, and 1 extra one. I'm going to move on to measurement methods. Here we have flow cytometry on the community level. This will show up in the data spreadsheet as community FC counts. We have strain level measurement for 16S RNA. These have been measured. These are relative abundances that have been multiplied by FC count. So arguably we could pick flow cytometry here or 16S RNA. It could go either way. This depends on the uploader to determine which technique description describes their experiment best. Ideally, this field should be filled in with an explanation. And this will show up in the spreadsheet as rRNA reads and RNA reads STD for standard deviation. Note that despite the fact that the spreadsheet column is called RNA reads, the units are in cells per milliliter. This might be confusing, but purely from a technical perspective, it's a little bit easier to match these up. We also have metabolite measurements of all these metabolites in millimolars. So this is fine. I'm going to click on next to move on to compartments. There's only one compartment. As a reminder, the compartment describes the environment of the experiments and all the experiments. Actually, there's only one experiment that is recorded that uses this particular compartment of Wilkins, Chager, and anaerobrot with a number of parameters filled in. We have a single community of all of these measurements and a blank community, which is here mostly for information purposes. We have a single experiment, which uses the full community, the WC compartment, and it has 6 time points. There are 6 biological replicates, biorep 1 to 6. I can edit this just so that we can review this a little bit better, but either way, we can move on. And now we get to the data template section. This time, because we have all the experimental information previously filled in, we can click on download template to get a template where we can fill in the data. I'm going to click on this button now, and I'm not going to save it for the moment. I'm just going to click on open with LibreOffice. You can open it with Excel or any kind of spreadsheet software that this format is compatible with. This gives me this following spreadsheet. There are 3 separate sheets for each of the types of technique. There is community growth, there is per strain growth, and there is per metabolite measurements. Per community, we only have the community flow cytometry measurements. If you hover on the cell, you can see the comment that describes these are flow cytometry values for every time point in cells per milliliter. There's also a hint to use period as a decimal separator. We have a time column, measurement time points in hours. We have a compartment column, which is the only compartment that we have in this experiment. I will later show you a different data spreadsheet with multiple compartments. And we have the label of the biological replicate from 1 to 6. That's pretty much it. The same thing we can see in the strain scenario. Note that if I scroll, the first row is frozen for convenience. And if I scroll horizontally, you can also see that the first 3 columns are kept. But you can resize them however you like. And one thing that you will see later is that the actual organization into sheets doesn't matter. What the system is going to look for is a column named in a particular way, and rows with particular bioreplicate, compartment, and time point. If we leave time points blank, they will be ignored. If we leave a column blank, it will not be recorded. To give you an example of how this might work, I have here a data spreadsheet that includes the raw data that I had for this particular study. And for example, what we can do is we can take the time points. You can see that the time points start for position A1. This is the first biological replicate. Start are located here. This is 6 time points. We can maybe copy those and paste them here. We can copy the Rosabora intestinalis reads and the standard deviation of those reads, and copy those, and then find Rosabora intestinalis in these columns. In this case, it is these 2 columns. I can paste these. Note that if these columns are formulas, you might get an error or something like this and get strange results. So a different thing you can do is paste unformatted text. In this case, this is not necessary, but this is also an option if you run into problems copy pasting from the Excel spreadsheet. It's a good thing to keep in mind that these values should align with the time points, to double check this, verify it, and so on. But with this, we can actually upload this spreadsheet with just this data, and we would actually get reasonable results. I've saved the spreadsheet as partial data, and I'm going to open it through this interface, through this browse files interface, and you can see that I get a preview of the data. You can see that the first spreadsheet, grow data per community, is shown here. No time points, no measurements. If I go to grow data per strain, you can see that there are time points in the first biological replicate, and you can see the measurements for Rosabora intestinalis. I'll now click on next, and this actually worked. We will discuss step 7 in the next video, but for now I'm going to click on this test study 1, and you'll see that there is some information on publishing. Again, we'll go into this a bit later. And there is one biological replicate. The other biological replicates are empty. They're still present here, but the only measurement here is of the bioreplicate 1 of Rosabora intestinalis. I can click on visualize, for example, to take a look at this and validate that it does in fact look as I would expect. So the purpose of this kind of partial filling in, to just fill in a couple of measurements, is to investigate and see if everything works as we'd expect. We can go back to the data upload. We can click on browse files again, and this time I'm going to click on template data field, which is the full data spreadsheet. As we can see here, with growth data per community, growth data per strain, and growth data per metabolite. If I now add this here, we will now see that this preview is updated. You can see that the file name has been updated here, the size of the file name, and we can review the different sheets here and see that this is in fact the completely filled in spreadsheet. Note that there can be gaps in it. For example, here we have an ethanol measurement that somehow it wasn't measured at time point 67. This is okay, this is acceptable. If you have missing values, it is okay to leave gaps. If you write 0 here, this suggests that there was an actual 0 measurement. So it's important to be aware of this. It's important to make sure that if you have missing values, they're simply not recorded in any way, either as NA or anything like this, and are simply recorded as a blank space. So we have this gets recorded here as not a number, so this indicates that it was a blank. If I now click next, again, we get the same sort of screen, but if I click on test study 1, you will see that there's quite a lot more information, quite a lot of measurements of bioreplicate 1, bioreplicate 2, and so on and so forth. I'm now going to show you an example with multiple compartments per biological replicate. I'm going to start a new study, very quickly call this one test study 2, test project 2, and then I'm going to reuse study 2 as its design with 2 microbial strains with optical density, community pH, and flow cytometry for strain level growth. So for community OD and pH, for strain level growth, we have flow cytometry counts and metabolites. Note that this is one example where we have 2 different types of metabolite measurements, one that is measured in millimolars and one that is measured in area under the curve. Particularly, these are the mucin sugars, which is why we add this optional label, and this allows us to upload the data for these metabolites with the optional label mucin sugars, whereas these metabolites are uploaded in millimolars. In terms of compartments, we have 2 compartments, the Wilkins-Chargren's anaerobe broth, and so that's the liquid broth in the vessels, and these are the mucin beads that we can measure separately. We have 2 monocultures and a co-culture. And for the experimental design, we can see, for now, let's just focus on experiment 1, BT mucin. You can see that we have 2 compartments here within the same vessel. So this is Blautia in a Wilkins-Chargren's broth plus mucin beads. And so the Blautia cells can attach themselves to the beads or detach themselves from the beads. And we have 17 time points to measure on them. And for the data upload, I'm going to show you what the final version of the data is. And one thing you can notice, for example, for the community level growth, we can see that we have the biological replicate BT mucin. Here are all the time points. And we have measurements for community OD and community pH. However, notice that we also have a separate compartment mucin with the same biological replicate. So BT mucin is now shown twice with different compartments, and that allows us to actually measure, upload these measurements for mucin as well. On the community level side for OD and pH, we do not have mucin measurements, but if we go to growth data per strains, flow cytometry counts are something that we can measure. In fact, here is the bacterius measurements for the WC compartment and for the mucin compartments separately. This is important to understand depending on your experimental design, depending on how exactly you structure it, that you would get these 2 separate compartments, one after the other. In most cases, this should not be the case. In most cases, you're probably going to have 1 compartment per biological replicate. Another interesting study here is study number 3, which I've created a new submission for, or rather I've copied its design. The experiment here, there's only 1 experiment with 9 time points. However, the raw data, if I were to click on the download template, this is what I get: community FC with 9 measurements, community pH with also 9 measurements. However, the raw data that we have actually has more measurements for pH at different time points. 70 measurements, and the time points don't even align. Now, one thing we could do is we could select a number of time points that fits the combination of pH and measurements here, and leave gaps between the measurements, essentially wherever it is that we measure pH and flow cytometry. But a fairly, like a more readable thing to do, and probably more easy thing to do, is to just create a new tab, a new sheet, and manually copy biological replicate compartment and time and community pH. Essentially, moving this column to a separate sheet and adding as many rows as it needs. I mentioned this before, I mentioned this earlier, but I do want to stress this, is that what you get here as a template is something that you can use. it is usable. But as you can see, this particular template has different fonts and columns because it's been taken from it's been copied from a different sort of data. None of this matters. What the system looks for is the name of the column and the specific information in each of the rows. If there's no data in this row for this column, nothing is recorded. Otherwise, here's your time point, here's your compartment, here's your biological replicate, and here is the column. This is something that works just fine if you do this manually in a separate sheet. In fact, what I did was I wrote some code that processes the community PH spreadsheet data files and produced this as a CSV and then opened it and pasted it in another sheet. This is an option if you have a bioinformatician on your team, if you're able to do this, but otherwise you can also just do this manually, create all these rows with the right labels for the biological replicate, the right labels for the compartment, and the time measurements for whatever it is that you're measuring. The final example I'm going to show you is an explanation of how we might process some data that is not organized in a reasonable way, or like a way in which fits what we request. For example, here is a spreadsheet with quite a lot of data, and we can see that the assay identifier is a great way to determine where this fits. So we have an experiment F5, F6, and F8, and then they repeat F5, F6, and F8. And this is a problem because if you want to now copy the data, we would have to maybe do it cell by cell. Ideally, it would be great to be able to just copy the entire column and then paste it into the spreadsheet. What we can do here is we can select this. One thing we can do is, for example, use the view to freeze rows and columns to make sure that this column is kept here. And we can select everything and apply an auto filter. And then for the assay identifier, I can type F5 to filter only experiment F5. And now for acetate, for example, I can simply copy this up until the end and then paste it in the actual spreadsheet. However, if we paste the values in the acetate column here and scroll down, we are going to notice that this last value is crossing this border here between the biological replicates, and this value is actually assigned to the first row of the next replicate. And this is an indication that something is off. Either the time points that we selected is incorrect, the number of time points, or there's something wrong with the data sheet. In this case, if we try to look carefully, we might notice that there are two rows for time point 80. And in fact, the values measured are identical, which indicates some kind of an issue with the formatting of the data spreadsheet. There might be some other reason for it if we investigate. But one thing that we can do for now is we can remove the extra row since both of the values are the same, and that way we can get, if we actually try to upload this duplicated data with another, like a duplicated time point 80, this would lead to a validation error. But it's important to understand why this happens, and in some cases there might be some kind of a problem when copying data that we notice that might not be caught by the validation mechanisms of the site. It's a good idea to use the time to transferring this data to the mGroupDB format to make sure that the data is uploaded correctly. After it is uploaded, it's a good idea to also go to the actual study and validate, evaluate the visualization and such in order to make sure that the data is actually representative of the reality of the experiment. But this is something that we'll cover in the next video.